Welcome to What's New in MPMI, the virtual seminar series of the MPMI Journal. I'm Jean Harris. I'm the editor-in-chief of MPMI, and it's my pleasure to have another wonderful talk by one of our authors here today. But before we get started, while people are coming on, why don't you type into the chat where you are coming from, and I'll read a couple off while we're waiting for everyone to log on. I see Sridhar Ranganathan is here from Chennai, India. Let me know where else you're from. Hi, Gary. Gary from Mizzou. Mike Colomedes from Texas. Sayan Chakraborty says hello. Sung Hwan from University of Missouri. Okay. All right. So while people, oh, Marco from Brussels, Prita Kundu from UNL, Cohen Gistling from Ghent, Belgium, and Jesse, oops, starting to go kind of fast. Jesse Kaleku from Ghana, but studying at the University of Wyoming. Miguel Aranda from Murcia, Spain, and Sayan Chakraborty from Kolkata, West Bengal. Okay. So, well, thanks everybody for joining. Always super exciting to see people, our colleagues joining from all around the world. I'm going to share for just a moment a slide of, we are not going to be having a What's New in MPMI seminar next month. And that's because IS-MPMI, the International Society for Molecular Plant Microbe Interactions, is hosting a wonderful early career showcase. Half of the talks will be in June 8th through 9th, and the other half will be in September. And what I have up here are just the speakers for our June showcase. And we have a lot of great talks coming from graduate students and postdocs, people, the new scientists in our community. And I'm delighted to see that we even have some on roots, my favorite topic, although of course leaves still dominate. This is just my advice. Go study some roots. Very interesting question. Anyway, I hope you'll be joining us for that. That's going to be coming up in June, and support our younger colleagues. Find out what's new. What is the new and exciting things that are happening? Okay. So, and I think that Karen is going to post into the chat how you can register for that, the link for that one. And I also wanted to let you know about, we have an upcoming focus issue for MPMI, the endomembrane system in plant microbe interactions. And I hope that some of you will be submitting your work. We have already, we have some excellent guest editors taking over for that. We have Hailing Jin, Dong Wang, Yunsuk Chun, and Aiming Wang, who are all going to be leading that. And I think that's going to be a really exciting issue. We have some great folks who've already committed to presenting that, but I hope you will. And we're going to post that link into the chat as well, so that you can join, so that you can consider, you can read about it and consider posting as well. Okay, and the last two things are just a reminder that that MPMI is having a special discount for papers with grad student first authors. So it's $800 off the cost of subscription. And we always like to encourage our early career scientists. So I hope that you will consider submitting to MPMI while that's still in process. That goes through the end of June. And finally, before I launch into today's regularly scheduled talk, I would like to ask for your assistance in helping to edit the transcript that we're creating as a part of this. Zoom does that automatically. I'm sure you've all seen that. Helpful, but not so helpful when it comes to personal names and to scientific terms. So if you would like to help edit that transcript and make our webinar more accessible to the greater community, this helps a lot with people who are hearing impaired in any way. It's also very helpful for people for whom English is not a first language. So I would love to have your volunteer help with this. If you're interested, please type your name into the chat, and we will be in contact with you. Okay, So now I would like to move on and introduce today's speaker, Debarati Basu. With me as co-host is Tim Friesen, who is one of our senior editors at MPMI, and he and I will be handling all the questions at the end. And while you have typed into the chat where you're coming from, and there's been some of the links for the papers, I want to remind you that all questions at the end should be posted in the Q&A, and you can post them at any time, and then Tim and I will take turns reading that at the end, and Debarati will answer. So I was very intrigued as soon as I saw Debarati's paper cross my inbox. I was definitely hooked. It takes us in a different direction than most of the papers we have in MPMI. So instead of effector interactions, reactive oxygen species bursts, this one is looking at the physical aspects. How does this cell sense these changes that are causing changes in turgor, or maybe changes on pressure outside the cell that can changes outside the cell that are physical forces? How are those sensed and interpreted, and how could that be one way that plants can sense that they're having an interaction with a microbe? And so it's just such a different approach, different way of looking at it. And talking with Debarati beforehand, I see that this was a different approach for her as well, and got her into some new areas. So Debarati Basu did this while she was a postdoc with Elizabeth Haswell, and she has now moved on to working as another postdoc at LSU, doing something other than Arabidopsis. So she's delighted to be working now with plants that you can actually eat. And maybe she'll give us a little bit of info about where she is now. With that, I'm going to turn it over to Debarati. You can share your screen now, and we will hear about your very interesting research. And I should point out that this is all based on this paper that is appearing right now in MPMI, and Karen will type the link to that in the chat, because of course, if you want more details, the best place to go is the paper itself. Thanks.
Thank you for that kind introduction. So I'm Debarati Basu. I'm currently a postdoc in LSU. I study photorespiratory bypass now in crop plants. So I will be telling you a story about a mechanosensitive ion channel and how it is involved in conferring plant defense against Pseudomonas syringae, a pathogen for Arabidopsis thaliana. So before we get into the nitty-gritty of the talk, I will walk you through the brief overview of plant defense response, the key players. So bacteria, the Pseudomonas, looks like this, and it enters the plant through natural openings or stomatas right here. This is a representative plant cell where I'm zooming into the apoplast, the area between the wall and the membrane. That's exactly where Pseudomonas will go, assemble, multiply, and then effect. So the basis of plant survival relies on promptly perceiving that there is a pathogen and then mounting a defense response. And the foundation of that relies on these receptors. Now, there are two types of receptors. One that are embedded in the membrane or called PRR, pattern recognition receptors. And then there are the cytoplasmic receptors, which are traditionally called NLR, the nucleotide leucine-rich repeats. Now, what happens is that this has to be very tightly regulated and inhibits defense response because it's a very costly process and the resources are limited under the condition where there is no pathogen. So in other words, don't think they don't want plants to be hypochondriac. Now, in response, when the pathogen goes into the apoplast under right environmental condition, they secrete this microbe-associated molecular patterns. These are characteristic conserved molecules that would be recognized by your PRR. The moment it recognizes, it triggers a downstream signaling cascades, and which we traditionally call pattern-triggered immunity, because it happens because consider these as ligands and these are the receptors, and plant confers its defense. Now, with evolution, pathogens have learned how to evade this interaction by directly delivering the effectors into the cell. So they are avoiding or bypassing these receptors. And they can do this by secretion systems. In Pseudomonas, that is a type 3 secretion system. It's like an injection that injects or delivers all the effectors, these proteins, right directly into the cell. Now, then what happens is that it relates to this effector triggered susceptibility because it inhibits PTI, which is detrimental to a plant. So plant also equally evolved this cytoplasmic receptors. What they would do is like they will bind to the effectors. This is a very simplified diagram. Just keep in mind the interaction can be very complicated, mediated by helper proteins, and trigger downstream cascades called effector triggered immunity. Now, with increasing evidence now show that although these are canonical players, there are also some surveillance proteins or immune regulators involved in immune response. For the interest of this paper, the most important of this is a protein complex, SGT-RAR1 and HSP90. These are 3 proteins together that are involved in ETI, and also 2 new proteins that are EDS1, NDR1. So they are neither NLRs or receptors, but they are still involved as immune regulators in defense response pathway. Now that we know this, we will shift gears and learn about these 2 terms that come over and over in the paper: Lesion mimic mutants and autoimmune mutants. What are those? So as I told you that this whole sophisticated, intricate process needs to be inhibited in absence of pathogen, but there are still mutants that show this spontaneous cell death. So in presence of pathogen, plants restrict the pathogen by activating localized cell death, which we traditionally call as hypersensitive response. Now, these mutants, when in absence of pathogen, can show a severe growth retardation and a spontaneous mutation, cell death. So these mutants are called lesion mimic mutants. There can be numerous reasons what leads to that. A subset of those lesion mimic mutants can be autoimmune mutants. Now, autoimmune mutants are those where the growth and defense, the fine balance between them, are disrupted. That occurs mostly by overactivation of any of their receptors, loss of negative regulation, disruption of this interaction of the given effector with its corresponding NLRs, or hyper or upregulation of salicylic acid biosynthesis, which is a key biotic stress hormone. And what would be the outcome of it? The plant will have severe growth retardation under ambient condition in absence of any stress, hyperaccumulation of ROS, spontaneous cell death or ectopic cell death, and constitutive expression of defense response genes like PR1. Now we will shift gears and introduce you to mechanosensitive ion channels. The question is, can mechanical signal triggers any plant defense response? Now, keep in mind, a lot is known about when the cells are severed, like by a herbivore. We are talking about where the cells are moderately intact, the wall is not broken, but still there is a membrane integrity perturbation. So these were the questions that we were having in mind. So now look at this ion channel that is embedded here. It's in the open configuration because of the changes in either type 3 secretion or the assembly of the secretion apparatus. That leads to a signaling cascade downstream. The question remains, can it use any canonical players of plant defense response? And then we can call it an autoimmune response. Just keep that in mind. So is it unique to the mechanosensitive ion channel? So what are the mechanosensitive ion channels that are already known in plants? So there are 5 classes of mechanosensitive ion channel that have been characterized in plants so far. And as you can see, they can conduct different types of ions. Today's talk is all about MSL or Miskase-like ion channels. They are found in bacteria and plants, but not found in animals. These ion channels are multimeric protein, and I would be talking only about the 10th member of the family. It's ubiquitously present in all tissues. It's localized in the plasma membrane. And we have shown through electrophysiology, the gold standard for characterizing any mechanosensitive ion channel, that when you increase the membrane tension by suction, there is a release of electrolytes. And then these ions that they conduct are non-specific. And they are slight reference to anions, and that's why I showed chloride. And this is a monomeric structure of the protein. And as you see, it has a very elaborate soluble N-terminus, which has 7 phosphorylation sites. And it also has an important site at the C-terminus, which we will come in a minute. So the first thing we started when we started studying this protein is that we looked at the knockout phenotype, and there is none. So we were forced to look into overexpression, even though we know that 35S can cause unintended effects. Now in 35S, you start seeing severe growth reduction and also this yellowing of leaves, which are nothing but spontaneous cell death that's detected by trypan blue staining, these blue spots. And when we also quantified H2O2 with DAB staining, we see those browning, which is nothing but hyperaccumulation of ROS. So then we were curious. It has a lot of phosphorylation sites, and phosphorylation are very important for triggering signal response. What about we make phosphomimetic mutants? Means take 4 of those phosphorylation sites detected by mass spec and then change them into aspartate or glutamate. That means they would be constitutively phosphorylated. And then you see that it reverts back directly to the wild type. Now you might ask if this is happening because the protein is degraded. The protein doesn't seem to be degraded. Even we detect higher abundance of MSL10-GFP in the phosphomimetic version, still the plants look like the wild type. Then we thought that what about phosphodiesterase, means change these phosphorylation sites back to alanine. When we did that, we were unable to retrieve a single viable plant, which made us to believe that probably it's a lethal mutant. While we were studying this, an independent lab also figured out a EMS-induced point mutation, which is a gain of function allele, which we named as MSL10-3G. And this is how the plants look like. It phenocopies the effect of gain of function or overexpression lines. Now, I have introduced a lot of new terms. Let's do a summary. In summary, this is an MSL structure. Keep in mind, this is a monomeric. It's actually a multimeric protein. In response to increased membrane tension, there would be release of osmolytes. But it also has a non-conducting function downstream that is mediated by the N terminus or the C terminus. If you overexpress this plant, it leads to cell death. Then if you have phosphorylation, that means under a normal condition, a wild type keeps MSL10 phosphorylated. That's why thereby no cell death occurs. If you overexpress this protein, you dephosphorylate them or have a C-terminus mutant S642L, you see the same effect, that is triggering of cell death. Now I will say the same thing with the help of images because that worked better. So these are my genetic tools I will be using in my that are used in the paper already. So this is wild type. This is a loss of function mutant. This is an overexpression, means you are making more protein. Then you have a point mutation, and then you have the phosphodiesterase. These three, that is overexpression, point mutant, and the phosphodiesterase are called MSL10 hypermorphs because they are activated MSL10 and they all lead to cell death. Whereas the phosphomimetic will revert it back to wild type. Now, what are the motivation that we want to even study MSL10 in relation to plant defense? Now, the spontaneous cell death that I have already shown in hypermorphic alleles resemble a lot of hypersensitive response. The other thing that is very exciting and why we study even Pseudomonas is that when you infect Pseudomonas, it stays in the apoplast where it is a requirement that it has an aqueous intracellular space. And technical term is called water soaking, that is required for its infection. This water soaking resembles a hypoosmotic stress. So previously we have published a paper where we have demonstrated that MSL10 triggers programmed cell death in response to cell swelling, which you can subject seedlings through hypoosmotic shock. And it utilizes a lot of downstream mechanism like a calcium transient that are very common in also autoimmune mutants. So that was our motivation of pursuing. So the very first thing we did is this question, can MSL10 serve as a membrane integrity perturbation sensor in response to Pseudomonas infection? So the first thing we did is live pathogen assay. We used DC3000, a virulent pathogen. These red stars indicate that only the hypermorphs, that is overexpression lines, phosphodiesterase or the MSL10-3G point mutant showed increased resistance because they have less bacterial counts. Whereas the complemented lines, the knockout mutant or the phosphomimetic lines look like identical to the wild type when you look at bacterial titer. So, and then our PAD4 SID2 is our positive control because it has already been shown to be more susceptible. Then we looked at the strain that doesn't have a type 3 secretion. So we had a mutant of DC3000 harpel. It will have an impaired type 3 secretion system. And we asked the same question, is it required for triggering a response? None of the lines that we tested have any difference from wild type. Then we traditionally tested the two avirulent pathogen, AVR-RPT2 and RPM1. In both cases, only the overexpression or the hypermorphic alleles only show increased resistance. The knockout mutants still do not show any phenotype compared to the wild type. So we started thinking that when I did these assays, I also should look into the PR1 expression, because I told you that constitutive PR1 expression is one of the hallmarks of autoimmune mutants. So I looked at the PR1 expression, and interestingly enough, unlike other autoimmune mutants, I don't see any constitutive expression under mock. So they look very similar to all the lines tested. Whereas when you infect it with DC3000, you start seeing an accelerated induction and also higher induction in the hypermorphic alleles that are labeled with blue, whereas the MSL10 knockout mutant has a very delayed response. It does get induced in later time points, but early time points, it's very reduced. So this got us thinking that probably MSL10 knockout mutant is a very subtle susceptible mutant. And I have to go back and look into the way I'm doing the assay. So initially I was infiltrating the DC3000, the bacterial strains. And I told you Pseudomonas needs to enter through natural opening. I am simply delivering the pathogen right into the cell. Maybe that's the problem. So I thought I will repeat this thing with two things being changed. One, I will do a spray inoculation. At the same time, I looked through the literature and found that this coronatine is produced by all virulent pathogens of Pseudomonas. It's a phytotoxin. What it does is that it forces the stomates to remain open so that more can come in and multiply. At the same time, it interferes with salicylic acid biosynthesis and signaling, because this coronatine molecule mimics a jasmonyl isoleucine, J active molecule. So I thought, why not use a coronatine-deficient mutant and then probe whether the knockout mutant has any issues with susceptibility. So as usual, I did first DC3000 because I want to compare spray inoculation versus coronatine-deficient mutant of Pseudomonas inoculation. Here only the gain of function line has increased resistance as seen before. But the moment we use the coronatine spray inoculation, you start seeing more bacterial counts in knockout mutant of MSL10, suggesting that probably these deficient mutant are having an issue, and then they are able to multiply more, and that's is the reason of increased susceptibility in the loss of function mutant. And then we also quantified the PR1 expression, and the same trend follows. The MSL10-1 has a very delayed response, even though there is an induction in PR1 in the early time points, whereas the gain of the point mutant, which is a gain of function allele, has a accelerated and a higher level of induction of PR1. Now, one of the reasons how coronatine can help the Pseudomonas to multiply is by opening, keeping the stomates open. So then we ask the question, is it possible that this higher susceptibility that we see in MSL10-1 is because the MSL10-1 is unable to close its stomates? So to probe that, I'm going to show you what happens naturally. So you put the leaves in a stomata opening buffer, the stomates, this aperture remains open. The moment you infect it with Pseudomonas DC3000 strains, it closes initially, and then because the strain can produce coronatine, it opens. This is done by exogenously adding coronatine, but the same thing happens when you have a virulent pathogen. And I will show you the data. So here at 0 hour, this is the stomatal aperture. In mock, there is no change. The moment you add DC3000, within 1 hour, the plant's natural response is to shut its stomates so that less bacteria can enter. But when you have a DC3000 strain, within 4 hours, it evades that response and then forces the stomates to open because it can produce this chemical coronatine. Whereas if you use a coronatine deficient mutant, initial there would be a closing, but only MSL10-1 tries to reopen it and performs better than the wild type. This suggests that maybe this is the reason why MSL10-1 is more susceptible. But at the same time, this would be in the back of your mind, you might be asking, what about MSL10's expression in stomach? I told you it's ubiquitously expressed. It's not specifically involved in stomatal closing or opening. To know that, what we did is that we used abscisic acid. We know that abscisic acid leads to stomatal closure. So if you add abscisic acid within 1 hour, you see stomatal closure, the same rate, the kinetics as that of all the lines. And within 2 hours, it further shuts down the stomates. This shows that the MSL10 doesn't have a developmental defect in stomatal closing and opening, but in absence of a mechanosensitive ion channel that could have sensed the Pseudomonas, it unable to shut it down as much as in response to coronatine defective mutant of Pseudomonas. So with this, we were curious that how can we probe the effect of the MSL10 hypermorphic allele and its ability to trigger autoimmune response. So if I summarize, I have Pseudomonas that we don't know how it elicits membrane perturbation, but as it seems like, then it leads to activation. This part we know because electrophysiologically we know membrane tension opens the MSL10 and activates its response. And finally, there are intermediate step that leads to ROS hyperaccumulation and ectopic cell death. So we looked into the literature. There are a lot of autoimmune mutants that are alleviated by growing them at elevated temperature. These are few of the examples. When you grow them at higher temperature, this small growth retardation gets sort of suppressed. So we thought whether MSL10 has the same thing can be true for MSL10 hypermorphs. So when we grew them at 21 degrees, this is how they look like. I'm only going to show you the MSL10 constitutively being expressed under 35S promoter. The same is true for all hypermorphic alleles as shown in the paper. So when you move them at 30 degrees Celsius, they all look identical. Now we quantified the fresh weight and you can start seeing that they are alleviated when you grow that at elevated temperature. Then the question comes, is it because the transcript levels are altered? Yes, it's slightly reduced at high temperature, but nothing compared to wild type levels. So it's not drastically changing the transcript levels to attain the wild type like phenotype. At the protein levels, it's even more interesting. There is more abundance of MSL10-GFP in the lines that look identical to the wild type compared to the lines that look very severely growth retarded. And finally, we looked at the subcellular localization. As you can see, both at 21 degrees Celsius or at 30 degrees Celsius, MSL10 localizes itself at the periphery. It's a plasma membrane protein. So we concluded that the suppression of the autoimmune-like phenotype of MSL10 hypermorphs is not because the transcript levels or the protein abundance or the subcellular localization alteration. It's something downstream signaling components are deactivated. So what about the cellular response like ROS and cell death? So we looked at the two flavors of ROS. One is superoxide detected by NBT and H2O2 detected by amplex red. In both cases, as you can see, there is the increase in their ROS is completely suppressed when you grow them at high temperature. Same is true for H2O2. Now in trypan blue, case of trypan blue, the same trend follows. You have a lot of cell death when you grow them at ambient temperature, but a complete suppression of them back to the wild type levels almost when you grow them at elevated temperature. Now, these are all related, associated to MSL10. What about the downstream hallmark genes that we have previously characterized that seems to be very critical for MSL10 induced cell death signaling? So these are genes that are already previously characterized in senescence, osmotic stress, biotic stress. There are 4 of them. So this was how it will look like under ambient, and we already reported that gain of function lines only have higher expression or induced transcript levels compared to the loss of function in the wild type. In this case, as you can see, this is wild type, this is loss of function. A huge induction of the transcripts at 21 degrees, but they go back to almost wild type level when you move them to 28 degrees Celsius. So this clearly suggests that this high temperatures by an unknown mechanism suppresses the downstream signaling component that are critical for MSL10 induced autoimmune-like phenotypes shown in the hypermorphs. So with this, we thought of looking into more details, how can we relate these phenotypic effects to autoimmune mutants? So this is what we have already seen. And this was the diagram that I was trying to put MSL into this whole players of plant defense response. Now we have issues. Neither MSL10 shares any structural similarity with the receptors, the PRRs, which are settled in the membrane, nor it looks anything similar to NLRs or cytoplasmic receptors. Then what are they? So one option would be the MSL10 is a target for the effectors, or it's like a GARDI. So to probe that, we looked at the very downstream components, the SGT, RAR1, and HSP90. So these three are protein co-chaperone complex or surveillance proteins that are involved in ETI, and then you disrupt them and you see loss of cell death in many of the autoimmune mutants. So that was our motivation. So we questioned whether disruption of three of these genes will alleviate the autoimmune-like phenotypes exhibited by MSL10 hypermorphic alleles. So what we did is that we took those three knockout mutants, we express MSL10-GFP in that background, then backcrossed it with wild type. And then this would be the wild type expressing MSL10-GFP. We did that to make sure that the genetic landscape is identical because the gene location is locked. Then we looked at their protein levels. As you can see, between the lines compared, there is not much difference in protein level. Where it is different, the line that produces severe phenotype does not produce a severe phenotype, has more protein than the line that produces a severe phenotype. So that's definitely not because the levels of MSL10 is different. Then we looked at the ROS and the tripe and blue staining for spontaneous cell death. In ROS, as you can see, SGT, RAR1, and HSP knockout mutant expressing MSL10-GFP has less ROS compared to the one in the wild type background. And the same is true for cell death. This suggests that these three genes are required for the MSL10 triggered autoimmune-like phenotype. So they act downstream of the MSL10 pathway. Now, the obvious question is, does that mean these three protein or the protein complex physically interact with MSL10 or not? To probe that, we utilize the very conventional protein-protein interaction assays, which include BIFC, biomolecular fluorescence complement assay. So these are my positive controls. MSL10 is a heteromer, so it interacts with itself. All these three proteins already have shown that they interact with itself. This is a negative control where MSL10 fused with split YFP does not interact with an empty split YFP. Now, when I introduced the HSP, RAR1, and SGT, none of them seem to interact with MSL10. We repeated the other conventional assays, split ubiquitin is2 hybrid, and both the assays infer that they don't physically interact. So we concluded that although SGT, RAR1, and HSP90 are downstream to MSL10 cell death signaling pathway and are required for the autoimmune-like phenotype, it does not physically interact. Although I'm showing you the one possibility we have not tested, and you should keep it at back of your mind, that there is a possibility that we tested them individually expressing them in tobacco. What about for interaction with MSL10? You need all the three proteins to be present, or at least two of them present. That we haven't tested. That is definitely a possibility. So then we thought of looking at the other immune regulators. Are they involved? So EDS1 and NDR1 are two immune regulators involved in ETI. So when it comes to ETI, you know that I told you that NLRs, or the cytoplasmic receptors, are required. There are two sets of them based on their structure. One are tall interleukin-like receptors, or coiled NLRs. The TLRs require EDS1 downstream to them, whereas the coiled NLRs require NDR1 downstream to it. So we had the same notion that what about we disrupt EDS1 or NDR1? Will that relieve the phenotypes associated with hypermorphic MSL10 hypermorphic alleles? So these are how the knockout mutants look like, and this is tripe and blue, and there is no cell death as expected. Now, if you express it under wild type background, you start seeing severe growth retardation and spontaneous cell death, as already shown. Now, if you express them in EDS1 and NDR1 knockout mutants, they look identical when it expressed under wild type. The cell death levels are also similar. So it seems that EDS1 and NDR1 are not required for the MSL10 associated cell death. These are their protein levels. The comparing line seems to have similar protein levels. This line does have less, but the loading control also shows that it has less protein. But I am always showing you in respect to overexpression, means MSL10 under 35 is tagged with GFP. But I also have shown that the C-terminus point mutation phenocopy is the gain of function. What about if we use that line? So this is MSL10-3G, the EMS-induced point mutation at the C-terminus, which phenocopies the gain of function right here. What about if I have EDS1 expressed in that? So this is an EDS1 knockout mutant and NDR1 knockout mutant expressing this mutant as a transgene. These are transgenic line. They look identical. This is an wild type plant expressing a transgene that has the point mutation. This is EDS1 knockout mutant expressing that point mutant as a transgene. So in both ways, the N and C terminus, we can show you that the EDS1 and NDR1 are not required for the MSL10 associated autoimmune-like phenotypes. So with this, the very last thing that remains is in that list of hallmark phenotypes exhibited by autoimmune mutants that we haven't tested is role of ASA. So what we did is that we have two genes, SID2 and PAD4. SID2 is a key SA biosynthetic gene and PAD4 is a transcriptional regulator for SA accumulation. We thought the same thing. If SA accumulation is involved in MSL10 associated autoimmune-like phenotypes, then we should knock them out and then start seeing alleviation. So this is a wild type plant. This is a knockout mutant plant, a double mutant. This is when you express 35S GFP in the wild type background, how it looks like. This is when you have a knockout mutant expressing 35S MSL10 GFP. Now you start seeing this plant is slightly bigger than the wild type, but not as big as the wild type without a transgene. So we quantified the fresh weight and we see the same trend. It seems like a partial reduction or alleviation of MSL10 hypermorphic allele phenotype, but not complete. And then we quantified the protein as well as we looked at the cell death. It's true that PAD4 SID2 may be required, but they only partially alleviates the phenotype. So with this, I come to a conclusion and I leave you with some lingering questions. So biotic stress by Pseudomonas syringae leads to elicitation of membrane perturbation by means that we don't yet know. Membrane perturbation activates MSL10. Then downstream to it is it requires SGT-RAR1 HSP90 co-chaperone complex, as well as a partial dependence on PAD4 and SID2. These all with some intermediate pathway leads to ROS accumulation ectopic cell death. Now that is the reason why it is resistant to Pseudomonas deficient in coronatine, accelerated PR1 expression, stomatal closure response. But the things that we do not know is that we need to know how to monitor actual changes in the amplitude of membrane tension when a pathogen is invading the cell. We do not know whether the flow of ions, because it's an ion channel, whether the flow of ions are required for MSL10 activation, because you can have a non-conducting function that is completely independent of ion channel. At the same time, we do not know how this activates ROS hyperaccumulation and ectopic cell death because it doesn't physically interact with MSL10. One possibility is that MSL10 acts as a scaffold where other interacting proteins comes and they actually trigger the downstream response. So with this, I will like to acknowledge my funding sources, all the members of Hashwell Lab, and I used BioRender to make my figures. And then I will be happy to take any questions.
Thank you, Debarati, for that very interesting seminar. Such an interesting set of questions. Let me take a peek. So this is a reminder that if you have questions, to please type them in the Q&A box. And our first one is a basic genetics question from Prita Kundu, who says, how do you define hypermorphs?
Right, so hypermorphs would be those which will have a gain of function phenotype. So they can be an overexpressing plant. I think I would do a better job in just showing you the image. I think image works better than just talking. Just give me 1 second. So my hypermorphs are, oh, sorry. Sorry. Yes, this figure will help you. So you have the overexpressor, you have the phosphodid, and you have the gain of function point mutant. These three are hypermorphs because downstream, they all do the same thing that leads to autoimmune-like phenotype. That's why we named them as hypermorphs. Does that makes it clear?
Yes, I think that's just another way of saying gain of function, really. It's in the sort of older genetic terminology. Tim, do you want to take the next one?
Yep, our next question is from Gary Stacy. What is the kinase that phosphorylates either the N or C termini?
So we don't know that. We spent a lot of time trying to look into that. I tried the conventional MPK3, 6, 11. MSL10 is notorious in interacting with anything and everything. Ingel kinase assay have some cysteine-rich kinase, but it's very inconclusive. So when you express one over the other, the phenotype you expect you do not see. But in vitro, it does interact or it phosphorylates. So we are in no way closer. So a graduate student is working in the downstream reactors by doing pull down assays and suppressor mutant. Stay tuned for her. She has some cool data to show. But I don't think we still know actually we need to know the phosphatase that dephosphorylates it because that activates. We are no way close to that.
Agreed. The phosphatase is a particularly interesting question as well, right? When you activate or turn off or inactivate. And just a reminder, if you're typing questions in the chat, to please type them in the Q&A so that we can answer them. While we're waiting for some more questions in the Q&A, one thing I was wondering is there's so many things that were surprises to me upon reading your paper. And I wondered, as you were going through the project itself, were there any surprises for you? Was there anything that particularly surprised you as you were going through this?
Yeah, absolutely. So the first surprise was that we didn't even pursue. That was surprising, and then we were like confused. Is that the paper that talked about the MSL10-3G, the point mutant, showed that that line has a lot of jasmonic acid. So they did the active molecule, the inactive molecule, the precursor. So I was always bogged down by the idea, how can you also need salicylic acid accumulation at the same time jasmonic acid, because Pseudomonas is hemibiotropic. So more jasmonic acid is detrimental because it will block SS signaling. That's the traditional thing I am more taught. And I know it is not that simple. It has more layers to it, but that was like very mind-boggling, which I never have wrapped my head around. Plus, we didn't think that stomatal closure would be anything to do because MSL10 is not specifically expressed in the stomates even. So that was also very confusing. And the other thing is that MSL10 is not a calcium channel. If it had been, I would be the happiest person. That stomates makes perfect sense. It's an ionic channel. So it has nothing to do with stomatal closing and opening when it comes to the channel function event. So yeah, that's a very baffling thing. We talk about that in our hypothesis as that it opens and the flow of the ion through the electrochemical gradient depolarizes the membrane, which can activate voltage gated calcium channels. That's our favorite answer to the reviewers.
I'll take the next one. Sung Hwan Cho asks, do MSL overexpression, does MSL10 overexpression show exactly the same cell death phenotype as the MSL10-3G?
It's exactly the same. Like we did try to quantify it with traditional other assays. So trypan blue staining, yes, it's a very similar levels of. Although it doesn't show the chlorosis of the leaf, I know why you are asking. The phenotype looks a green leaf, but it does have the same level of cell death.
So then the next question is Mike Colomites. Sorry if I got that wrong, but his question is about salicylic acid levels in the overexpressor lines.
We don't know the levels because we never quantified. What we can only tell you that because disrupting the PAD4 SID2 does partially alleviate, I would assume that there is a higher level, but whether that higher level persist all through the plant's life or not, I don't know for sure. Or is it like a transient when in the vegetative state it's growing, or it's specific to the leaves, or there's a systemic response? We don't know any of those.
Jesse Kaleku asks, do you know if the MSL10 and its mutants have an effect on the expression of NLR proteins?
No, we haven't looked, and we were hoping to pursue that. The moment I can show EDS1 or NDR1 are required, then I was ready to jump in. But also remember the AVR, RPT2, RPM1 didn't, like the knockout mutant didn't show any increased susceptibility. So if the gain of function and loss of function do not have the positive effect, there also, I don't know, we can look into RIN1 protein or anything of those. I was very prepared to look into all those things, but because EDS1, NDR1 were not required, so we did not pursue that line.
So one of my questions, Debarati, was thinking about this at more of a whole leaf level. Where is MSL10 expressed? And right, you have, I'm curious about whether this sudden efflux of anions is also affecting cells sort of in a cells that don't express it, right? So that it could be a non-cell autonomous kind of effect triggering. So
it is ubiquitously expressed everywhere. We have looked at roots and leaves through like traditional GAS. It doesn't stand out any different in any cell. It doesn't get induced upon, like we looked at MSL10 induction in response to Pseudomonas infection. There is no induction of it at the transcript level. So it's all at the protein level or the downstream signaling component through which it's signaling. It's not at the transcript level. There is a question here from Gary Stacy about what is known about how mechanical stress activates this channel. So when you say mechanical stress, we do electrophysiology, where I can just share a picture. I knew this thing, it's not very traditional. Right here. So this is how we look into, why can't I get to the right slide? Sorry. This is how we characterized ion channels. So in mechanosensitive ion channels, you express them in root protoplast or frog egg cells, Xenopus oocyte, and then you have this threne tube where you pinch. That pinch is actually a single protein. It's as narrow. It's under microscope. You can't see it. Then what you do is like you start seeing the wild type, basal level and the mutant. And then when you suck it out from the cell, then you start, because there's the suction creates the tension and you starts increasing of channels. So these nice step-like things are different ion channels. This is the current it's flowing because there is a flow of electrons through it. Flow of ions through it, and this is closing event of the channel. So this is what we know, and this is done in completely either root protoplast or the heterologous system. But you realize we can measure the tension that we are putting it or the negative membrane tension, but we don't know in a live plant how much tension is there. That part we do not know. So these are expressing a protein or the wild type or a mutant. This one is more native compared to the heterologous experiments, I get it. But we don't know how much tension in a plant is required for the gating of MSL10 in vivo. That we do not know. And then protoplasm would be much different than a leaf epidermal cell where everything is like a puzzle shaped. And so it won't be very, you can't sort of extrapolate that same tension. It would be in the ballpark, but we don't know that.
What do you think is just speculate, because of course you don't know. This is part of the thing, one of the things that you said, we don't know. What do you think might be causing that membrane tension? What aspect of infection?
Right. So there can be two things. One is that the type 3 secretion, because when we have the HARP-L, none of them seems to have any effect. Or it might be just the load of the pathogen. And that's where we do not know exactly like how many pathogens are like the titer required to induce a downstream reaction. The other complete indirect method has nothing to do with those local membrane tension. It's all about the water soaking, which might look like a cell is swelling because more water is rushing into it for giving space for the Pseudomonas, and that triggers MSL10, which has nothing to do with biotic stress. It's just assuming it's like an abiotic stress of cell swelling. The outcome of those things are the same, but how it's happening are very different. So that might be another effect.
What about, go ahead, Tim.
So do you think this is bacteria specific, or do you think this is across other pathogens, oomycetes, fungi?
We haven't done any experiments, but we will love to. But when you start looking at other fungi, especially the one that have Haustoria or Apressorium, that will be a direct relation, like where it's physically imparting on the membrane. Plus the chitin would be important, where you are chewing the cell wall, and are you making the cell wall more weaker, where the membrane tension would be more. We don't know that, and we will love to try some fungal pathogen. Oomycetes would be the ideal because they infect like Arabidopsis. Many of the ones that we want to try don't even infect Arabidopsis.
Yeah, I was definitely wondering about the cell wall aspect of it. If you could, as such a key aspect of infection, breaking down the cell wall, changing the cell wall, whether that could change things. What about roots? You said that it's expressed ubiquitously. I don't know anything about these lesion mimics, what happens in roots, but I am assuming there's some responses down there as well. What do you think might be the most interesting aspect to follow down there?
See, that would be cool to look at. So our hypoosmotic stress, everything is done in roots because it's easy to image, and it's like chlorophyll. So all the dyes that are in the same wavelength as GFP, autofluorescence works wonders. I don't know, like Pseudomonas does not infect through roots as much as I know. It's a phyllosphere specific pathogen, but there are those Pseudomonas, other strains of Pseudomonas that are isolated from soil. They might be cool to show, but I always envision root are under perpetual tension and stress because they are facing boulders and soil particles and whatnot. So I just don't know how you will differentiate both. And in an assay, if you grow them in agar, you are taking that away. So mimicking it in a lab experiment would be difficult. But yes, wounding in roots would be a place to start. Like if you wound, and do you see an induction of MSL10, or like how does the overexpression like effects? That would be nice to see, but I think it's just the challenge of designing an assay where you have a live pathogen. We can't use elicitors or anything. We need the live pathogen to create the membrane tension, if that's the hypothesis we are going. That would be just tricky. But that's a cool way to think how in roots it works.
Yeah, a lot of questions following up on that, but I'll turn it back to Tim.
So yeah, we have another question from Preetha Kendu. They say, is there any known role of MSL-10 in herbivory?
Oh, this is interesting. So there is a science paper we collaborated where they, it's not herbivory, but they chop the leaf. So assume herbivory leads to severing of the leaf. So in that, it's shown that MSL-10 acts with the glutamate receptor GLR for long distance electrical signaling and calcium waves. So that came right when we were working. So I helped them in the electrophysiology part of it. So it seems like it may be because wounding can induce and jasmonates are important. So yeah, that's another aspect of looking at it with the caterpillars and the live herbivores.
Yeah, in roots, I was thinking of a similar thing with the, some of the like root knot nematode type things. I think there must be an Arabidopsis system for
Yeah,
those.
I think we have some collaboration with the nematode that was like the cyst forming one and then a non-cyst forming one. It's very strange. Again, another strange data. Apparently, they leaves MSL10 because they need that tension to move like whatever adults and then the larval stage. So it's very confusing. I think we collaborated with Melissa Mitchcomb. Am I saying it correctly? So their lab, but it's all those collaborations where we don't have any clear idea what's going on.
Yeah, because I was thinking those giant feeding cells, that there must be all this huge swelling in there. Anyway, so many questions. Do you have any more questions yourself, Tim?
No, I don't think so.
Well, I think, as we wrap up here, I thought there was another question coming in. What advice, I know you said when we were talking earlier that this was a really new step for you going into this area. And what advice would you have? I know we always have a lot of graduate students listening for taking these kinds of steps in their career.
I would say, I know it's very intimidating and you feel like, what if you fail? But then the joy and the rewards out on the other side, it's much more. And then you always think like you will always have this fallback idea, right? There was a new field, I tried my best and I failed, rather than I always learned how to do Western blot, I'm still struggling with it. So I think you get that motivation also. And it's very rewarding if you can push yourself through it. It's really, it's almost those hiking or like some sort of physical thing where you don't push yourself, but once you do it, it's very rewarding. And then you will learn a lot from the failures also. That I think I learned in a very hard way, that I always expected things to work and then most things do not work and rarely they work. That seems to be the case, but then you will get used to it. That ups and downs are part and parcel of grad school and postdoc. But I think that's also the reward, that those highs and lows. And then you will also learn to collaborate with people. Asking for help is also something you have to learn, because with more knowledge, you tend to feel that you should know everything, but that's not the case. And as a student, that's the best time to ask questions. Nobody's going to judge you. That's the learning phase. So you are in the perfect scenario and situation right now. So take up as many new challenges, new instrument you can. So it's just not for building your CV, it would just emotionally will help you to tackle more academic challenges.
I agree. That's what makes science interesting, is constantly learning something new. Certainly for me. Anyway, thank you so much, Debarati, for an excellent seminar. Very exciting. Love to have this as a part of our What's New in MPMI. And just to remind people that you can check back to the same place where you registered for this seminar and find the recording. And that should be up in a day or two. And hopefully at that point, we will also have an edited transcript to be able to include. So yeah. Thanks very much.
Thank you so much for the opportunity. Bye.
Bye, everyone.